Okay, this lecture is about cell membranes and the purpose of cell membranes and also how things will pass through cell membranes. So there's two types of cells that we'll talk about in this course. There's prokaryotic cells and eukaryotic cells. Both prokaryotic and eukaryotic cells have cell membranes. And we'll talk about the outer cell membranes, and then we'll also talk about some inner membranes of organelles. We also will talk a little bit about osmosis, which is the diffusion of water across membranes. So the cell membrane is the boundary of the cell, but it is not a static structure. It's dynamic and it's constantly moving. These are some pictures of some kind of unique looking cell membranes. So cell membranes are flexible. It allows the cells to change shape and also move through smaller spaces. So blood vessels, for example, are small, and the cell membranes and shape of these red blood cells here are able to kind of squeeze through these small openings because they have these flexible cell membranes. The main component of cell membranes are phospholipids, cholesterol, proteins, and carbohydrates, but mainly phospholipids. So if you remember back to when we talked about the different classes of macromolecules, one of the classes were lipids. Lipids are fats. And a special type of lipid is called a phospholipid. So this is a picture here of a phospholipid, and you see it has two parts. It has the hydrophobic head and then the hydrophilic tail. And the hydrophobic head is made up of a phosphate group, and that phosphate group means that it is happy to be around water. The other part, the hydrophilic tails, where these two tails are, these tails do not like water. They are called hydrophobic. So you might have seen examples of things that don't mix that are hydrophobic and hydrophilic. A good example of this are oil and water. So you know, if you put oil and water together, they don't mix. They just separate into two separate layers. Water is hydrophilic and oil is hydrophobic, so they don't mix. So kind of the same thing is happening with these phospholipids. So this is a picture of some phospholipids showing that they can have different sizes and shapes and also different compositions of the hydrophobic and hydrophilic parts. Polar and nonpolar are also ways that we can describe these different regions. Water is polar. You'll learn about this in chemistry, the structure of water because of its structure and the angles, the separation of charge. It's polar, and then the hydrophobic parts are non-polar. So this is an example of a phospholipid bilayer. So you can see this cross section here. It's called a bilayer because there are two layers of phospholipids where the hydrophobic tails are facing each other and the hydrophilic heads are facing either the outside of the cell or the inside of the cell. Cells are aqueous, meaning they are mostly made up of water. So you want the part of the phospholipid that's facing the aqueous water environment to be the hydrophilic part, which is the head part where the phosphate group is. So this is kind of the classic model of a membrane. It's called the fluid mosaic model. So a membrane is not like a smooth surface. It would be kind of like if you had a swimming pool and you filled it up with all different types of floaties and balls, and it covered the whole surface of the pool. If you allowed it to sit, it might look like it was one solid structure, but as you moved things around, things would move and kind of float around. So that's what's happening in a cell membrane. We have what's called this fluid mosaic model. We have the phospholipids that make up the majority of the lipid bilayer, and then we have other things that are integrated or inside the membrane. So we have cholesterol, proteins, carbohydrates. And you can see all of these. We have what are called integral membrane proteins. An integral membrane protein means integrated. It's going through both sides of the lipid bilayer. You also have what are called peripheral membrane proteins. A peripheral membrane protein is just on one side of the membrane, so it's not spanning both of the lipid bilayers. You also see these cholesterol molecules. Cholesterol is really important in cell membranes. It helps maintain plasticity, fluidity. You want just the right amount of cholesterol in membranes. No cholesterol is bad, and too much cholesterol is bad. So there's a specific amount of cholesterol that different cell membranes need, and also the type of cell membrane it is. So if this is a cell membrane in a type of fish that lives in the Arctic Ocean, it's going to have a different composition of cholesterol in its membranes than a warm-blooded mammal would. So the fluidity and the correct composition of the membrane is maintained by the amount of cholesterol that's in the membrane. You'll also see some carbohydrates on either side of the membrane. A lot of these carbohydrates and proteins on the outside of the membranes are how cells communicate with each other. We don't talk about antibodies in this class particularly, but oftentimes you would see antibodies or receptors or things like that on the outside of cells. Membranes are what are called selectively permeable, meaning it lets some things in, but not all things. So it can prevent harmful material from entering and also prevent essential material from leaving. So it is a good barrier, but it does selectively let things pass in and out. And there's two major types of transport in membranes, and that's called passive and active. Passive transport is sometimes called diffusion. Diffusion is always moving from a high concentration gradient to a low concentration gradient. And it means it just passively passes through the membrane. It doesn't require any energy. It doesn't require any membrane proteins. It doesn't need any help. It can just go right through. So here's the lipid bilayer. Gases such as oxygen and carbon dioxide and other small non-polar molecules can freely cross this lipid bilayer, again, from areas of high concentrations to low concentrations. But other solutes, which are other particles that are dissolved, cannot cross the lipid bilayer on their own, so they need some help. This is showing an example of diffusion. It's showing how when this dye is put into the sample of water, and the molecules will start to diffuse until they are evenly dispersed among the water molecules. So in diffusion, the molecules always move from an area of high concentration to low concentration until equilibrium or homeostasis is reached, which just means equal distribution of whatever that substance might be. Another example, well, a specific example of passive transport is osmosis, which is just specific to water. So it's diffusion, but just for water. And the movement of water from a low solute concentration to a high solute concentration. So in this example, if we have this 2% solution here, and it is in this picture here where it says isotonic. Iso is a word that means same. So that means it has the same concentration of sucrose on the outside as it does on the inside. So it's the same, so it's just equal concentration, equal moving back and forth. Hypertonic means, hyper means more, hypo means less. So hypertonic means that the concentration outside is higher, which means water is going to come from this bag of the 2% sucrose, and it's going to leave, which is going to make it all shriveled up. So this is kind of representing what would happen in a cell if this was a cell with this 2% sucrose. Hypotonic means that it's a lower concentration outside, so water rushes in to try to equalize the concentration. So we'll look at examples of this. We'll do some homework questions and also some of the exercises in the lab that we'll do will illustrate how osmosis works. So this is an illustration of that using red blood cells. So here's isotonic. Iso means same, meaning that the concentration is the same inside and outside the red blood cell. Hypertonic means that the concentration outside is higher. So that means water is going to leave the red blood cell, leaving the red blood cell all shriveled up. Hypotonic means that the concentration outside is higher and the concentration outside is lower, the concentration inside is higher. So water is going to go into the red blood cell to try to equalize those concentrations, and the red blood cell would swell and maybe even burst. And here's some examples of plant cells being isotonic and hypertonic. Same idea. Some species can also have what's called osmoregulation. So this is regulating the level of salt and water in your blood. Fish are in a hypertonic solution, seawater, so the concentration of salt in the seawater is higher than the concentration of the salt in the fish. So they're at risk of losing water or having high salt concentrations. So they have special adaptations for this. Their gills and other systems prevent this. So this is another example of passive transport. We're still going down our concentration gradient, but we need a carrier protein to do it. So we're using the protein, the transport protein, to go through. And what makes this passive instead of active is that there's no energy required because we're still going down the concentration gradient. So you see there's more glucose in the extracellular fluid than there is in the cell in the cytoplasm. So because we're going down the concentration gradient, this is still considered passive transport. We just need a transport protein because glucose cannot just passively diffuse through because it's too big. This is just showing how the transport protein changes shape to allow the glucose through. So active transport is moving against the concentration gradient. So you can kind of think of this like a pump, like it's going against the concentration gradient, or kind of like going uphill. It's going from a lower concentration to an even higher concentration. Active transport requires energy, and it uses energy in the chemical bonds of ATP to pump this, whatever it is, in this case it's ions, potassium and sodium, against their concentration gradient. Another example of active transport is called endocytosis. Endocytosis is when the cell membrane will kind of grow around something. This is called phagocytosis if it is a large particle that it's like eating. Phago means eating. Or it can be called penocytosis, which is where it's mostly just taking in water and ions. Peno means drinking. So cell eating or cell drinking. And this is where the membrane kind of grows around whatever is outside the cell and brings it to the inside. This is something we see a lot in microbiology and something we'll continue to see in this course. So that's that for membranes and different types of transport, and I'll leave it there.